So, with the very first class for Cambridge O Level Biology 5090, well, in fact, this one's for O Level and IGCSE, but because we only have a Cambridge student, right, that's why I have this particular syllabus open. Uh, First of all, you need to know your subject code. So it is Cambridge O Level and the code is 5090. So that is for Cambridge O Level Biology. So you need to know your subject codes. And going over the syllabus is the first thing I do whenever we begin classes, a series of classes for each session. Uh, these are the aims that Cambridge says it has with the syllabus. So it should enable students to acquire scientific knowledge and understanding of scientific theories, develop a range of experimental skills, use scientific data and evidence to solve problems, communicate effectively and clearly using scientific terminology, notations, and conventions, understand that the application of scientific knowledge can benefit people and the environment, enjoy science, and develop an informed interest in scientific matters. That is important. The enjoyment part, I believe, is the most important. This is an overview of all the topics in the syllabus. There are 19 of them. We will try and cover all of them. That is my target. We are starting classes a bit late in the session, and so I might have to speed through some of the topics, focusing only on the salient points, the main points, and right summarizing the content. So it is highly advisable that you read the chapter whenever you can after classes or before classes if possible. That's even better, right? So you're able to keep up. The assessment overview, so there are three papers that you would have to give. Paper one, by the way, this is the syllabus for 2026 to 2028, right? So there are three papers you would have to give. Paper one is a multiple choice MCQ paper. There will be 40 questions, each question worth 1 mark. The total time you will have is 1 hour. Paper two, the theory paper, 80 marks, 1 hour, 30 minutes, which gives you 1 hour, well, 90 minutes, right? So that's pretty much the Cambridge standard, which is 1 minute per mark. So that's pretty much it. Over here, you've got more time because you have to mark your question answers on the question paper and then transfer them to an answer sheet. And then either paper three or paper four, not both. Generally, here we do paper four, which is the alternative to practical. 1 hour paper, 40 marks, right? And this will test experimental skills, right? So paper one MCQ, paper two short answer and structured questions, and paper four in this case would be testing experimental skills. The weightage for each is 30, 50, and 20, which brings you to 100. Then we've got assessment objectives. So there are three categories of objectives that will be assessed. A01 is the very basic, which is assessing the knowledge with understanding of a student. So they should be able to demonstrate knowledge of scientific phenomena, facts, laws, definitions, concepts, theories. Then scientific vocabulary, terminology, and conventions such as symbols, quantities, and units. Scientific knowledge of scientific instruments and apparatus, including techniques of operation and aspects of safety. Scientific and technological applications with their social, economic, and environmental implications. Right, so this is mostly basically everything that you will get in the syllabus in the textbook. Right, students subject content defines the factual material that candidates may be required to recall and explain. And candidates will be asked to ask questions which require them to apply this material to unfamiliar contexts and apply knowledge from one area of the syllabus to another. So that will become clearer when we go through the syllabus and later on when we are doing past papers. Assessment objective 2, handling information and problem solving. So candidates should be able to, in words or using other written forms of presentation such as symbols, graphs, and numbers, locate, select, organize, and present information from a variety of sources. So you've got graphs or tables or text with information in it, and you're supposed to be able to pick out relevant information. Translate information from one form to another. So maybe you have a graph and you pick out, you read the graph, you get the numbers, you explain that. So manipulate numerical and other data. So you're using uh, values again taken from a graph and then you plug them in perhaps in a formula or something else again, right? These assessment objectives are the same pretty much for the sciences. So you'll find the same assessment objectives for chemistry and physics as well, right? So there might not be as much, in fact, there isn't as much of calculations to do in biology, but then you have got chemistry and physics where perhaps this is applied in greater to a greater effect. Use information to identify patterns, report trends, and form conclusions. Present reasoned explanations for phenomena, patterns, and relationships. So you're reading a graph and they're asking you, well, why is it the way it is? And so you explain, oh, it's because so and so process was occurring, XYZ. Make predictions based on relationships and patterns, and solve problems including some of a quantitative nature. So questions involving numbers. Questions testing these skills may be based on information that is unfamiliar to candidates, requiring them to apply principles and concepts from the syllabus to a new situation. That is the highlight of Cambridge. They put you in new situations, right? And so you should take on concepts and apply them, even though they might name something that you're not familiar with, but the concept still applies in a logical, deductive way. So in case of biology, they might tell you about an enzyme you've never heard of. But the questions they'll be the situation that they'll present is where you would need to apply uh, your knowledge about how enzymes work, for example. Right? So even though it's an unfamiliar enzyme, you know what would happen if the temperature changes, for example. Assessment objective 3, this is all of this is only limited to the practical or the alternative practical paper, right? So AO1 and AO2, you'll find them in both paper 1 and paper 2, so the multiple choice and the theory paper, whereas AO3 is only in the practical paper. So AO3 is 100% of the practical, and you will not find AO1 or AO2 over there. That information is also given on the next page. So what about experimental skills and investigations? Candidates should be able to demonstrate knowledge of how to select and safely use techniques, apparatus, and materials, including following a sequence of instructions. So the paper might give you instructions to follow for an experiment, and then the questions based on that. Plan experiments and investigation. This is perhaps the question that students find most difficult. Although in reality, if you practice it a bit, it actually isn't. Right? Uh, so planning questions. Make make and record observations, measurements, and estimates. Interpret and evaluate experimental observations and data. Evaluate methods and suggest possible improvements. Right? And so we'll experience this is something we'll experience when we get to paper 4, right? But it will be based on everything that we do through the syllabus. Right, so here's the weightage. In total, out of 100% of your papers, AO1 has a 50% weightage throughout, AO2 has a 30% weightage, and AO3 only 20%. That actually corresponds to the weightage of the papers. I believe. Right? Not mistaken. Yeah. Individually, through the papers, you can see that paper one and paper two both have a 63% weightage of questions that are of the AO1 category and 37% of the AO2 category, none of the AO3, and you can see paper 3 and 4, so you're doing only one of those, but both of these have 100% of the AO3 category. So those are the kind of questions you'll get. Now, coming to details of the syllabus, right, the subject content. Now, I'm not going to go over each topic in detail, but I want to highlight is that the syllabus is a very useful document. So you can see over here that uh, they've given you the name of the chapter, cells. That's the very first one we'll be getting into. And then, right, for cell structure and function, and then specialized tissues and organs. Those are the two subtopics of the chapter. And then they've given you a list of things you need to know. Right, so there are two ways this is helpful. One, right, is knowing exactly what you need to know. Right, this is the kind of information you like. If you know this, you know the syllabus. And on that note, you can also help this, like, so this is a checklist. Do you know this or not? Right? If you know this, you've covered the chapter properly, you've covered the content properly, you're good to go. The second benefit is as quick revision. So you can see over here, for example, that, right, not so much in these chapters. For example, point number 3 over here in 1.2, you're revising, oh, what's the formula of magnification, where it says over here that you need to know, you need to be able to state and use the formula for magnification, which is image size over actual size. And so not only is that, right, something you need to know, but it's also revision for you, right? So that is the formula. Or, right, any of these. So investigating factors that influence diffusion limited to surface area, temperature, concentration, gradient, distance. So can you explain these? Yes, that's a checklist. But it's also quick revision. Oh, what are the factors that influence diffusion? Surface area, temperature, concentration, gradient, distance. These are the factors that influence diffusion. So quick revision. Right. So skipping all of that, you've got details. Seems rather lengthy. Oh, we're not done yet. There we go. Right. Are we back there? Okay. Uh, skip. All right. This is of relevance. So over here, the practical test and alternative to practical require the same experimental skills to be developed and learned, require understanding of the same experimental contexts, test the same assessment objective AO3. Okay. And this is the part which, it seems very long, but you need to like at least know that this is something you have, and also, right, uh, you make good use of it. So candidates are expected to be familiar with and may be asked questions using the following experimental contexts. Right? Simple quantitative experiments, including measurement of volumes of gases, masses, temperatures, lengths, times. So you need to be able to, in the alternative to practical, you need to be able to tell how to do that and actually perhaps make some calculations. Uh, right? You need to be able to explain diffusion, osmosis, and tests for food, the rate of enzyme catalyzed reactions, pH, and the use of hydrogen carbon indicator, litmus and universal indicator, photosynthesis, along with rate, factors that affect the rate, and factors that affect the limit the rate of photosynthesis, effect of mineral ions on plant growth. So you could be given a situation where that is being tested, and then anything related to it. So a relation perhaps, right, plant growth, and then they would, right, link that to photosynthesis, and then they would link that to perhaps testing for food or diffusion. right? Or obviously transpiration is there, right? So germination maybe, right? So they could link it to other topics. So transpiration could be tested, heart rate and breathing, respiration, tropic responses, nervous responses, observation and dissection of seeds and flowers, germination, continuous and discontinuous variation, sampling techniques, use of a microscope to examine biological specimens, calculating the magnification of biological specimens, procedures using simple apparatus in situations where the method may not be familiar to the candidate. Yes, you have a question. Well, these are the different experimental scenarios that may be presented and tested in the paper, right? So you can see that these are all topics that are covered in the syllabus as a whole, right? But which of those topics might show up in your practical or alternative to practical paper? So they're giving you a list that these are all the topics, right? So they're not entire chapters, they're basically uh, subtopics or concepts that may be tested in paper 3 and paper 4. All right? Yeah. Okay. Candidates may be required to demonstrate knowledge of how to select and safely use techniques, apparatus, and materials. But this one's important, right? So I wanted to have procedures using simple apparatus in situations where the method may not be familiar to the candidate, right? So if you know how an apparatus is used, if you know how something is tested, then even though the situation is unfamiliar, you can try and work your way through the experiment. Okay, keep in mind they're testing your understanding of how experiments can be conducted. Okay, so where were we? Yes, how to select and safely use techniques, apparatus, and materials. So you need to be able to identify apparatus from diagrams or descriptions, draw complete or label diagrams of apparatus and biological specimens. Right, the drawing of biological specimens is another highlight of paper 4. Use or explain the use of common techniques, apparatus, and materials. Select the most appropriate apparatus or method for the task and justify the choice made. So, why are you using the apparatus or equipment for a particular test? Describe food tests. Describe tests to determine the pH of solutions. Describe and explain hazards and identify safety precautions. Describe and explain techniques used to ensure the accuracy of observations and data, right? So, how can you make sure that your results are as good as they can be and as close to what they should be in theory? Planning experiments and investigations, right? So identify the independent and dependent variable. Now this, right? We're just starting off, and this might seem like a lot. Oh my God, right? There's so much of it. Well, as we cover, all of these things will be highlighted, and then when we come to practice, right, we'll get into obviously applying these things, and so right. But yes, there is some technical details that we need to cover. So, what are independent and dependent variables? We will talk about that. Describe how and explain why variables should be controlled in experiments. Suggest an appropriate, and this is, yeah, so suggest an appropriate number of range of values for the independent variable. Suggest the most appropriate apparatus or technique and justify the choice made. Describe experimental procedures, identify risks and suggest safety precautions, describe how to record the results of an experiment, describe how to process the results. So recording the results, you might want to record the results as a table. How will you process the results? Well, will you make a graph or a pie chart, or what will you do with it? To form a conclusion or to evaluate a prediction, make reasoned predictions of expected results. Right? This is actually a guideline for your planning paper, planning question. Sorry. Right? This is pretty much what you have to do in the planning question. Where did we go? Okay. Make and record observations, measurements, and estimates. So take readings from apparatus. Now, obviously, you're not doing it. You're not doing paper three. So paper four, they might give you an image of some sort where you have to read off a scale or the stopwatch or something, right? Whether it's analog or digital, uh, or from diagrams of apparatus with appropriate precision. So they might give you a zoomed in uh, image of a measuring cylinder, for example, and you will have to read the volume of the liquid in it. Observe and take measurements from biological specimens or images of specimens. So this is more like, you know, magnification questions, basically, where they give you an image and they'll ask you to measure this, the length or the width of whatever it is the image is about, and then, move on to calculate the magnification or something. Take sufficient observations or measurements including repeats where appropriate. Record. So an experiments just taking one value is not enough. Oh, I did the experiment once, this is the result I got, and that's that. No, right? You want to take a range of measurements, right, over a range of values, right, of the independent variable. Uh, for example, you are measuring the rate of enzyme activity over a range of pH. So you're not going to take the rate of enzyme activity at just pH 5, for example, right? You will do it over a range of pH and see how it goes, and then you will repeat the experiment at that same pH at least 3 times, right? And that would make your result reliable, that you got the result you got the first time is what the result should be. So the results you get for the second and third repeat should be close enough to the first one, right? Otherwise, if it's very different, then that would highlight an anomalous result, so an outlier, right? There's something wrong, there's something went wrong, and that's why this result is very different from the other two, for example. So you need to repeat the experiment at least 3 times, right? Uh, if the experimental setup allows for repeats. Record qualitative observations from food and other tests. So changes in color, changes in size, maybe, right? Those are qualities. Uh, Record observations and measurements systematically, for example, in a suitable table to an appropriate degree of precision and using appropriate units. So units, what are you measuring? Are you measuring something in, right? If you're measuring the weight of seeds, then you probably be using grams. You're not going to be using kilograms because seeds are not heavy, unless you're talking about coconuts, but then you won't be doing that. And to an appropriate degree is, right, normally I like they're talking about significant figures, so that's something. And what works is 2 to 3 significant figures, or, right, the same number of significant figures given in the question. Depends. If they haven't identified anything, unless they ask you specifically, give your answer to 3 significant figures, then you do that. Interpret and evaluate experimental observations and data. So it seems like a bit of a repeat, but process data including for use in further calculations. So you calculate one thing, and then 2 steps later they're asking you to use the data the answer you got in part A3 in part B2. So yeah, that happens. Or for graph plotting, so you might be asked to plot a graph using a calculator as appropriate. So yeah, you can take that gadget with you. Present data graphically, so you should be able to do that. Analyze, interpret observations and data, including data presented graphically. So you talked about that. Use interpolation and extrapolation graphically to determine a gradient or intercept. Right. So what is interpolation and extrapolation? So extrapolation is you go beyond your studied points and predict what according to the trend, right, of how what behavior could you perhaps notice if you went further on in the experiment. Whereas interpolation is, give me a sec. Right, so interpolation is the process of estimating unknown values that fall between two or more known data points. So, for example, you've taken measurements after every 5 seconds or 10 seconds, and you've got values on the axis for time as 10, 20, 30, 40 seconds, and they ask you to find the value at 35 seconds. Well, you did not measure that, but you have all the other values. So you're trying to, you use the graph to find the value at 35 seconds, which obviously you did not uh, get during the experiment. Okay, where are we? There we are. Form conclusions justified by reference to observations and data with appropriate explanation. Right, so if you're making an observation, it should be justified based on whatever data you have. Straightforward. Right, you can't just throw something out there and say, well, yeah, that's what I think happens. Evaluate the quality of observation data, identifying any anomalous results and taking appropriate action. So I talked about that. Right, if you're given a set of data and they're asking you, well, which data point will you not use? So you need to look at it carefully and see, oh, well, this one looks different from the others, so I won't use it because it might be anomalous, it might be an outlier. How will you fix your data set? Well, you might not use that value, or you might repeat the experiment for that particular setup and conditions. Evaluate methods and suggest possible improvements. Right, so evaluate experimental arrangements. Are they good enough? Methods and techniques, including the use of controls. Right, something to compare your results against. Identify sources of error. Suggest, right, the source of error is not like, oh, I made a mistake. That's not a source of error. Right, you're going to identify either the limitation of your experimental techniques or the limitation of the apparatus, or a practical problem that you were having perhaps. Right, not that it's not, oh, oops. Suggest possible improvements to the apparatus, experimental arrangements, methods, and techniques. How can you make things better? Okay, now this I'm not going to read in detail. Apparatus, materials, and reagents. Here's a list of, right, biological materials, chemical reagents, right? So a selection of prepared slides of plant and animal tissue, locally available terrestrial and aquatic plant material, yeast. So these are things, right, that you might come across in the paper. Again, if there was a practical, then you would might have to deal with these in the lab. For the alternative to practical, there might be questions about these. Chemical reagents, right? So all the different reagents that you would need to know and perhaps talk about uh, in your ATP, right? What you need to know, what agar jelly is, what the Benedict solution is, what dilute acid or dilute alkali is, right? What hydrogen peroxide solution is, how is it used, and by the way, the symbols given, right, tell you about their safety precautions, right? And so I believe they're given somewhere later. Yeah, no, because I do not remember these by heart. Right, C is corrosive and MH is mildly hazardous, right? So anyway, so right, these are all the reagents that you might need to talk about. Obviously, all the not, are not going to come up in the same question or the same paper. So this is a bit of information base that you will develop through the syllabus and through practice. right? All the apparatus that you need to be familiar with and perhaps be able to use. And yes, right, you should know what a balance is that can measure up to 500, right? To be able to select for your experiment a balance that can measure up to 500 grams, or electric fan, right, for checking the effect of wind speed on transpiration, for example, right? Or glass rods or heat proof mats, tripods, and causes, microscope, obviously, right? Scissors. So you might need to mention that in some experiment, right? Stopwatch, syringes, wash bottle. Okay, still a bit left. There are mathematical requirements. Right, so you need to deal with numbers directly, add, subtract, decimal fractions using standard form, percentages, use decimal places appropriately, use significant figures appropriately, algebra, so, or algebra, as they say, right? So algebra, substitute values of so algebra is basically finding unknowns, right? So using known terms to find unknown terms, and direct and inverse proportion and all of that. Geometry, so understand the meaning of angle, curve, circle, radius, diameter, circumference, square, rectangle, and diagonal. Recall and use the equation of the area of rectangle, area of triangle, and area of circle. So you need to know those formulae. Recall and use the equations for the volume of a rectangular block and volume of a cylinder. So you need to know those. Understand surface area and use surface area to volume ratio. Using scale diagrams, select and use the most appropriate units for recording data and the results of calculation. So that's something done. Converting between metric units, including micrometer, millimeter, centimeter, meter, centimeter cube, decimeter cube, milligram, gram, and kilogram. And using mathematical instruments such as a ruler or a protractor. Graphs, charts, and statistics. So drawing charts and graphs from data, interpreting charts and graphs, including interpolation, so and extrapolations, talked about that. Determine the gradient of a line, determining the intercept of the line of a graph on a graph, extending the line graphically where appropriate, so basically reading the graph and finding unknown values. Select suitable scales and axes for graphs. How will you fit in your data points in the space provided for the graph? Recognize direct proportionality from a graph, right? So linear relationships, if those are what you're getting. Use and interpret Venn diagrams to show groupings and sets. I haven't come across these as much in biology, but perhaps. All right, calculate the mean and range of a set of values. Use simple probability. Presenting data, so taking and recording readings. Right. Just a bit more left to do. Taking and recording readings, so data should be recorded so as to reflect the precision of the measuring instrument, right? So uh, if you're using a meter rule and the smallest measurement you can take is, right, 1 millimeter, for example, then you're not going to give your value as right, 36.74 centimeters, because the 7 up till 36.7 makes sense. Okay, fine. So there's 36 centimeters and 7 millimeters. But where did the point 0.04 come from? How did you figure that out? Because the scale is not accurate to zero point 0.1 millimeters. It doesn't give you that value. So data should be recorded so as to reflect the precision of the measuring instrument. That is the smallest difference that can reliably be detected on the measuring instrument scale should be reflected by the number of decimal places given in the measurement. So just explain that. A measurement or calculated quantity must be accompanied by a correct unit, right? So you just can't write a value. If you're writing the length of something and you write down 63.7 and you don't write centimeter, for example, and so that means nothing. 63.7 watts, 63.7 kilos, 63.7 meters or centimeters, 63.7 seconds. It could be anything, and therefore, without a unit, your number means nothing. And so, right, you need a proper or correct unit where appropriate. So, certain cases, such as right, where you don't need a unit, such as the gradient, for example. Okay. Each column of a table should be headed with the name or symbol, actually both. Sorry, name or symbol and unit. So name or symbol of the measured quantity, right? And the appropriate unit. So time is the name of the quantity and S is the unit, right? Second. Now this is, right, the solidus, that basically that slash is to be used for separating the quantity and the unit in tables class. So this is how you will represent whatever values you're putting into your table under appropriate headings. Units should not be included with data in the body of the table. So you've put it up over here in the heading. And so whatever reading of time you're putting here, you don't need to put in 5 seconds, 10 seconds, 15 seconds. No, you've put up time in seconds in the heading. And so everything else you're writing down, we know you're talking about seconds. You don't need to repeat that with, right? The number of significant figures given for a measured quantity should be appropriate to the measuring instrument used. Again, right, similar to what we talked about here. Graphs. The column headings of the table can be directly transferred to the axis of a constructed graph. So basically, right, the values would represent what the x and y axis on your graph would be. The table headings, sorry. Uh, the graph should be drawn with a sharp pencil. The axis should be labeled with the name or symbol of the measured or calculated quantity and the appropriate unit. Unless instructed otherwise, the independent variable should be plotted on the x-axis, which is the horizontal axis, and the dependent variable. All right, so just to clarify, what is the independent variable in experiments? So, for example, you want to measure the uh, rate of uh, transpiration, maybe rate of water loss, or rate of photosynthesis, right? And you put up an experiment. So what you're measuring is, so what the unknown is the rate of transpiration or the rate of photosynthesis. What are you going to do to measure the rate? So for transpiration, I might use an electric fan and set it at different speeds to cause movement of wind, which would cause a higher rate of water loss, which is basically what transpiration is. So my independent variable is the variable that is under my control, which is the fan speed. The dependent variable is the rate of water loss, which is happening due to my variable. So the fan speed that I'm changing is affecting the rate of water loss, right? So that is what independent and dependent variable means. So what we're saying over here is independent variables should, right, unless otherwise instructed. If there are specific instructions in the question, you follow those instructions, you forget everything else. If there are no instructions, then the go-to is plotting the independent variable on the x-axis and the dependent variable on the y-axis. Independent variables are the parameters that I am changing in the experiment to test something. So taking a different example, I am changing the light intensity to test the rate of photosynthesis of a plant. Okay. Now we know that light is required for photosynthesis. Right? It's one of the requirements. So, right. because light intensity is under my control, that is what I'm changing to test photosynthesis. And so the thing that is under my, the variable that is under my control, the variable that I am changing in the experiment is the independent variable. Okay. And then whatever it is I'm testing that is being affected by the change in light intensity, which in this case is the photosynthesis that I'm testing, right? And it is the dependent variable. Why? Because the rate of photosynthesis depends on the light intensity that I am providing. Okay. All right. Unless instructed otherwise, the scales of the axis should allow more than half of the graph grid to be used in both directions, so vertically and horizontally. Right, so using at least half of the graph grid, at least, right? In fact, allow should allow for more than half. Sorry, not at least half, but more than half. And be based on sensible ratios. So 2 centimeters on the graph grid representing 1, 2, or 5 units. So make it easy to use, right? Don't put in such a scale that would be difficult to process. So you're right, you know that 2 and 5 can work together, right? 1 and 2 and all these, right? You're putting in something like 7, and that would become a little tricky to read off the graph, or putting in 3, right? So, right, points on the graph should be clearly marked as a cross, and a cross is not like a huge cross, like a small cross, just to mark the point, or encircled dots, right? Dots of appropriate size. You don't want like a fat dot, like a blotch on the graph paper, right? Because the point of data is actually, very accurate. And so you're just putting like a pencil dot over there and circling it perhaps, or putting a cross over it, but that's it. You're not going to make it thick, just so that you know, you say, but it's easier to read. No, don't do that. Each data point should be plotted to an accuracy of 1/2 of one of the smallest squares. Right now, this would actually make more sense if when we are dealing with the graph. So I'll leave that for the moment. Uh, a best fit line should be a single thin smooth line or curve drawn by inspection. So, right, the line does not need to coincide exactly with any of the points. So, best fit means it should pass through 3 points at least. No, maybe it's not passing through any of the points. Right? Where there is a scatter evident in the data, examiners would expect a roughly even distribution of points on either side of the line over its entire length. Right? So best fit line basically means that you are seeing the distribution of points and the trend, and then you're going to draw either a straight line or a curve depending on how the data points are going. The line or curve does not need to pass through any, does not need to, if it is passing through any of the points, well, sure, but don't try to adjust the line so that it passes through a particular number of points. And also, uh, right, if it's not passing through any of the points, then right, try and make it such that there's just about an even number of points above and below the line, right? Because that would qualify as best fit. Points that are clearly anomalous should be ignored, right? So if there's a point lying way outside all the others, that's not going to be considered for your best fit. Candidates should be able to take readings from the graph. We talked about this. Direct data value should be read from a line on a graph to an accuracy of 1/2 of one of the smallest squares. Okay, the same accuracy should be used in reading of an intercept, so either the y or the x-intercept. Drawings, these should be drawn using a sharp pencil, fine lines, clear and unbroken, no overwriting, no thick lines, no heavy lines, Uh, these should there should okay these should use most of the available space and show all the features observed in the specimen with no shading. So you're not going to color or shade or anything, but you need to observe the image properly and then, right, go through it. Label line should be drawn with a ruler and touch the object or feature labeled. Now, what I'm going to do right now is I'm going to show you a paper 4 in specific after I'm done with this. So we're almost done. In fact, I'll show you all three papers, just so that you know what is all of this talking about. Okay, because if you don't know the papers and you haven't gone through the book, then right, this would not be making a lot of sense. Pie charts are generally used to show percentage or proportional data. Bar charts should be drawn from categorical or discrete data. They should have bars of equal width that do not touch. Right? Histograms should be drawn for continuous data. They should have bars that do touch. Okay, decimal marks. Decimal mark examination papers will be a single dot on the line, right? Numbers, variables, I think we can skip this. Units should be the SI units, right? Approved SI units. So you're not going to write something in inches or Fahrenheit. Command word. So these are all the words that might show up in your papers. And then this tells you how to answer, right? What the question is expecting of you. So this is given in alphabetical order, not in the order that they would appear. So what does, if the question says assess, that means make an informed judgment. So you probably have some data or some information based on which you're going to give an answer. Calculate, so work out from given facts, figures, or information. Comment, an informed opinion, right? So based on what you know from your syllabus or maybe data that you've been given. Compare, identify or comment on similarities or differences, right? So comparing means similarities and differences both. Contrast, this is basically commenting on differences, right? Uh, Define, give precise meaning, right? They might be asking you for certain definitions of what things are, right? So, for example, what is a tissue? And so you give a precise definition, precise meaning of what a tissue is. Describe, meaning, right? What you need to state the points of a topic or give characteristics and main features. Now, how this actually applies to a particular question, right? That will obviously come from attempting those questions and from experience. Determine. So, establish an answer using the information available. Okay. Discuss. Write about issues or topics in depth in a structured way. So, organization is important. And you need to go into a bit of detail, so in depth. Evaluate, judge or calculate the quality, importance, amount, or value of something. Explain, set out purpose or reason, make the relationship between things clear, say why and or how, and support with relevant evidence. Give, so that's basically produce an answer from, information that's given or from memory from the syllabus. Identify, so name, select, or recognize, pick something out basically. Outline, set out the main points, just the main outline, give the main points, no details. Predict, prediction, state, so I suggest what might happen based on information available. Sketch, make a simple freehand drawing showing the key features, taking care over proportions. State, express in clear terms. Okay, now we are done with the syllabus. Are there any questions regarding the syllabus itself? I know you probably have questions about right, some of these things and their applications. So for that, I am opening up, right, the specimen paper for the current syllabus. So that's from 2023. And let me open up paper one and paper two and paper four. Okay, so these are specimen papers. Yeah. So this is multiple choice. There's not much to talk about here. Some of the questions. So each question will have four options to choose from, right? The four options are A, B, C, and D. So they might give you some other information, right? Such as one, two, three, four, all the way up till five or six or something, right? But the options that you need to select for your answer is either A, B, C, or D, right? So which structures are found in animal cells and plant cells? You got mitochondria, yes. Cell walls, no. Not in plants. Animal cells, ribosomes, yes. Sap vacuole, no. Right? So this is 1 and 3. So that's A. Okay. Now we're not going to solve this. Just that was just an example. This is a magnification, so you need your ruler and protector and sharp pencil. Also, you need your proper equipment for the paper, right? And you need your calculator as well. And you can see that, well, the actual is 0.008 millimeter, right? This is 20 millimeter. You don't have to measure it, but you do have to calculate it. So you need your calculator for that. Uh, it can look scary, but you need to read the question carefully, and you need to read the information carefully, and then pick out. So this is a question from classification. uh, and then, well, you get the idea, right? Some questions are simpler than others. Over here, right, this is an experimental setup, and they're going to ask you, well, right, which one does what, or which one is the correct setup, perhaps. uh, yeah, so that's paper one, right? Identifying parts or uh, you know processes or hormones or whatever it is. Okay, paper two, the theory paper. Zoom out a bit more. Oh, by the way, uh, you would notice in paper one, they've given you information on the front sheet. Always read the information that's given to you, always read the instructions that are given to you, even though you know what they are, right? Still, don't skip them. But you see over here, there's nothing given for your name or candidate number, center number. That's because this is not the answer sheet that will be checked. This is the question paper where you will be marking your answers. And as I told you, you will have a separate answer sheet where you will mark only the A, B. So it will have, right, question one and circles for A, B, C, D. And then, right, it says over here, uh, write in soft pencil, and right, for each question there are four possible answers. Choose one you consider correct and record your choice in soft pencil on the multiple choice answer sheet. So you mark it over here. Whatever scribbling you need to do, you do on the question paper for your ideas or information that you're recalling or calculations. But the final answer that you selected, you circled it over here, put a tick mark, or however. Use a pencil, don't use a pen, right? Pencils are cleaner, so you use a pencil here. And on the answer sheet, use a pencil only. So you circle, for example, one was A, and you see, okay, question one, A, B, C, D. You circle A only, and you circle, you shade, you fill in the circle A, and you fill it inside only. So don't go outside, and don't be creative or sloppy, either case. Over here, because your question paper is also your answer sheet, so you're answering on the paper, and so you've got space for your candidate name, center number, and candidate number. So you'll fill those in, right? You can see 5090 is your subject code, 02 is for your paper. Over here, it's given 02 because the specimen paper, it will most likely have 2.1 or 2.2, right? So that's paper 2 variant 1 to paper 2 variant 2. Over here we have variant 2, so it will be 22, 5090 22. These are the, right, you have different types of questions, some simpler, right? These are like fill in the blanks. You've got, right, a describe question over here. So going back, right, what are you being asked for? Describe, state the points of a topic or give characteristics and main features. So that's what they're asking you over here, right? Describe what happens to these amino acids after digestion until they reach the liver. So give the main points of the process of digestion and what happens up until reaching the liver. Here's a graph, so you'll have to explain the trend. You'll have to read from the graph probably, right? And explain. So talking about fertilizers and plant crop growth or something. Uh, More fill in the blanks. This is for genetic diagrams and Punnett squares and all that. So a photomicrograph of a pollen grain with a pollen tube growing from it. Okay. Some information and then some calculations perhaps, some details you're trying to figure out depending, like based on the information that they've given. So lots of parts. Oh, and by the way, in these questions and also in your ATP, remember that the general rule for Cambridge is one valid point per mark. Generally, not 100% absolutely. There are certain questions where you need to write two things for 1 mark, right? So those kind of questions you will also pop up. But generally, right, so if it's a 3 mark question, you need three valid points, right? This over here is a 4 mark question. Earlier on, you had a 6 mark question. And so, right, 1 mark means one valid point. Okay. Fill in the table, right? Seems like you're back in kindergarten or something, but yeah, it's not, perhaps not as easy as it looks. All right. So, anything out of the ordinary? All right, so you can see you have all of these command words coming up, outline and state, right? That's what I wanted to show actually. I'm just skipping through this. So you got lots of states over there. Uh, Compare, right? So similarities and differences is what you're going to do. Describe another, describe one. Suggest, right? So all of these are coming up, and so right, the command words tell you what is expected and how to approach the answer. Explain, and yeah, we're done. Now, paper four, right, which had so much information in the syllabus regarding paper four. So let's see what we have over here. So first up, right, they're giving you an experimental situation regarding uh, reducing sugars apparently in Benedict solution. So you can see include a suitable safety precaution in your answer, right? Describe how you would use the solution. So how you would use the solution, a safety precaution, 3 marks. So two uses and one safety precaution. That's how it would work. Sorry, two steps of the procedure. How you would use the Benedict solution. So two steps of the procedure and a safety precaution. You can include more because in this case they are not telling you how many points they want. So if you write a bit more, it doesn't hurt. Okay, so this is based on experiment and they're giving you some data and then they're asking you questions based on that. State and explain and describe and all of that, right? And suggest. You got a blank page. Blank pages can be used, right? They're not to be left blank. So if you do calculations or make some notes or something, you can do that on the blank page. Here you'll be asked to make a graph, right? In this case, they're asking you to make a bar chart, right? So the bar charts, equal widths of the bar and lines not touching each other. So there should be a gap in between them, right? This is the information you have to make the bar chart over here. So you need to, right, figure out how wide each bar should be to cover more than 50% of the area of this graph and represent the data properly. And then they will ask you to read the bar chart and give information, right? State and suggest and all of that. 6 marks for, right, Planning. So this is a planning question, right? 6 marks, planning question, whatever the thing is, the testing for vitamin C content. So yeah, right. Here is the magnification question, I believe. Yeah, and this space is to draw. So all the sketching, right? You look over here, you see the details, right? Proportionality. So what does it mean by proportionality? Right? So I'm just zooming in. Obviously, this kind of zoom is not available in your actual paper, but you see over here, for example, the shape and angle of this cell, right? Or whatever this is, and then the shape and angle of this one, and then this is the smaller one over here, G, right? So the position of the smaller one, the shape of the smaller one, the size of the smaller one as compared to the larger one should be accurately represented in your own drawing, right? You cannot change the proportions. So obviously you're going to make this larger in your drawing. This one, it's already like large over here. You're going to make it larger, so you make it something like that. But as big as you made it, this one will also be bigger, but compared, it should look the same way. Okay, I don't know if that's making sense to you at the moment. Is it? Yes? No? Are you still there? Okay. Is it making sense? Kind of. Okay. So magnification, drawing, describing, and then, yeah, that's the end of the paper. So that's what an ATP is like. So I just go to the whiteboard briefly to just draw something to explain to you, right? Uh, how proportions are maintained, and then we'll end. First class a bit lengthy, but I didn't want to leave the syllabus for the next class. So from our next class, we can get into covering the syllabus. So what I meant by proportion is, right, the cell that you saw. Okay, let me go back to the question. Where is it? All right. Right, so what the question is saying over here, let me put that on screen again. So what the question is saying over here is draw the starch grains. So these are starch grains labeled E, F, and G. So there are three of those as they appear in figure 3.1. Grain F, so the large one in the center, should be at least 60 millimeter long, right? And so that means that you will, obviously use a ruler and you will mark 2 points, right? Here and here perhaps, right at two ends. I don't know if you can see me touching the screen. And right then, right, the extreme ends, so the tip on both ends should be at least 60 millimeters long. That's 6 centimeters. So you use a ruler, you make it slightly larger, the space would allow probably, right, going from one end to the other. So if I can just access my whiteboard, should be something like this, right? You use a ruler and you make the starch grain. Are you able to see my whiteboard? No, I haven't put it up. Okay. There's the whiteboard for you, and over here And then, okay, going back. So this is what it will look like, right? So that's F. And what I meant by proportion is, right, I think it was G, it was over here somewhere. So I think that's a bit too, so like that maybe. And the other one was somewhere over here. So even though the ones I've drawn are bigger than the ones in the picture, which is the whole point, but right, they still seem to be, in comparison with the large one, they still seem to be, the same size difference. It's not, it's not given micrometers. Where is micrometers given? Well, right, they said 60 millimeters. So when you're using a ruler, right, 60 millimeters is 6 centimeters. So yeah, but right, so this is perhaps 65 millimeters. So you use a ruler and you mark 2 points. Okay, this is how big I'm going to draw it. And then the shape. Now I haven't been very particular, but the shape, right, should be as given in the paper. So let me just go back to the paper. So yeah, so oops, too big. Okay, so the shape of F, right, this guy, and right, the shape of G, and E, and their position and their you know relative size to each other, or how big or small they are compared to each other, that should be maintained in your drawing over here in the space provided. All right. Obviously, when we get to it, when we actually do that, it would make more sense. I believe, right, we've had a very nice, lengthy session, but we've covered the syllabus and its requirements, the syllabus document, and everything you think you need to know. You've looked at the papers, and so from the next class, uh, we will start with the actual syllabus content. As I mentioned, because we're starting a bit late, I might rush through certain easier topics. And so, right, if there's anything confusing or unclear, you're obviously free to ask, because that's the whole point. And in any case, keep up with the textbook. So, reading the content on your own. All right. So, I'll see you next class, inshallah. Allah Hafiz.